Yeah, ladies and gentlemen, welcome to the afternoon session, which we started a little bit late, but when it comes to high performance techniques, sometimes we experience some problems, so my apologies that we are a little bit late. But I hope that you will enjoy very much the session this afternoon, which is about the growing role of HPC in neuroscience. And it is my pleasure to introduce this project to you and also my colleague. So the Human Brain Project is a project that is trying to understand, to contribute to better understanding of the human brain. And I would like to change to the second slide. Oh, now we have it. Okay. And when we are looking back, then neuroscience and brain research are very closely related to each other for a long time now. As you can see here from a drawing from Leonardo da Vinci, who did this draft of the human brain, probably not most of his most beautiful activities, but he also was a big inventor and also created a first draft of a calculating machine. So at least in his head, neuroscience and computing were very closely related to each other. Can I have the next slide, please? This was further continued when we think about the 50s of the last century, where HEP and other neuroscientists found out what are the rules of neuronal networks. And only a little bit later, the first version of the perceptron, which was one of the first artificial neuronal networks, has been developed. The next slide shows that, of course, along these lines, we have different developments. And so, for instance, we have published just recently a very high-resolution reconstruction of a human brain on the one hand, and on the other hand, we have large European initiatives driving the development of new computing technologies. Next slide, please. So in that sense, the Human Brain Project only brings together what has been together for a long time. And the Human Brain Flagship project aims at a better understanding of the human brain as one of the most challenging research topics, not only of the 21st century, I think. And it is so important to understand the human brain because it helps us to understand what makes us human, but also very practically from an economic point of view, it helps to drive the development of new technologies, and it helps also to develop better therapies for patients, which is really one of the goals that we have to address. And we are now starting the next period of our funding of the so-called phase SGA2. So now I would like to have the slide. So why to do it in such a large project and not in only a small research group? This is not visible, it's very white. Ah, here we are, okay, now it was good. Thank you. And the reason is that the brain is so complex, and if you think we are communicating, our brain is a couple of centimeters long. If we are addressing small structures like the hippocampus, we are coming down to the millimeter and micrometer level. And within the hippocampus, which is so important for learning and memory, we see that there are many, many thousands of cells being located, which are connected through so-called axons. They form little networks. And information is transmitted from one neuron to the other using so-called synapses, which are very small and tiny structures, a few micrometers large. And in these synapses, we find vesicles with transmitters, and these transmitters are crucial molecules for signal transduction. And transmitters bind to these receptors, which can be a few nanometers or even angstroms. And to understand, of course, how all these different levels of organization works together, this is really a challenge that needs a very large project. And therefore, I would like to introduce my colleagues from the Human Brain Project to you, which give you some insights into simulation, but also in big data analytics. And if I may finish, then I would say our flagship project is not so much a flagship project like Vasco da Gama had this a couple of years ago, but perhaps it can be transferred into a more modern and advanced form of a flagship. Okay, also does not work. I'm sorry, so we have today trouble with our technique, but it's indeed an enterprise to approach the human brain to better understand it. So I would like now to hand over to my colleague Jeanette Kotaleski, who is a professor at the famous Karolinska Institute and who is doing research on simulating networks. And this is also very close to medicine because mechanisms that she is analyzing are very important for signal transduction. So, thank you very much for joining us here.
Yeah, so first I would like to thank the organizers for inviting me. Next slide. I will try to exemplify what it would mean to simulate the brain at one level, and I will just start where Katrin what she already mentioned, that why do we want to understand the brain? Well, it's pure curiosity, and we also know that brain damages and brain diseases accounts for 1/3 of the healthcare cost in the Western world, and therefore we need to understand the brain at a mechanistic point of view. And if we understand the brain, we can also extract new principles for computing, and we capitalize on all these things in the Human Brain Project. Some sort of well-known facts about the brain is that it is an electrochemical organ, which have 100 billion neurons, and every neuron connects to other neurons at several thousand connection points called synapses. And recent theoretical research sort of has predicted that every synapse might store up to 5 bits of information, which actually theoretically could mean that the storage capacity of the brain could be in the order of the pages on the internet. Still, our brains run on very little energy. It just needs approximately what a traditional light bulb needs. And I think also we contemplate far too seldom that everything we know about the universe, all of these fascinating theories we have, it's a product of the information processing in our brains. And now we try to understand our own brain. So in this context, I would like to cite Sidney Brenner. He's one of my favorite scientists. He says that progress in science depends on new techniques, new discoveries using these techniques, and then new ideas, probably in that order. And when it comes to the brain, the current techniques allows us to collect an enormous amount of data at different levels of organization. And there are some challenges, and that is that the brain field is sort of a bit fragmented. A lot of different disciplines are really interested in understanding the brain, but data is collected from different points of views. And this, of course, means that we have a lot of facts and a lot of neural correlates at different levels of organization, but the real challenge is to integrate these and create knowledge and understanding from the data. And then one way to do that is to use modeling and simulations. And we have heard already that simulation-based sciences have really sort of revolutionized many fields. And of course, a challenge is that the brain is very complex and very much multi-scale. But anyway, if we have something like the brain, which has many dynamic, complex, and interactive processes in parallel, at some point we need to simulate it on various scales. And yeah. And it's quite obvious that no single lab can simulate the brain themselves. So it has to be really a collaborative effort. And this means that to be able to collaborate, we have to agree on how to model and simulate the brain, and how you validate results, model results, and so on. And in this context, it's also quite interesting to point out that even though we have a lot of data, this data is actually sparse. If we want to simulate the brain at the cellular level, it means that we have to represent all these 100 billion neurons and their connections, but we will not be able to measure those things. So a very important part of building models of the brain is to find the principles and the algorithms that can actually live with sparse data and predict what is missing. So this is an important part of the modeling workflows. So before I will try to illustrate what we do in Stockholm, I should also just highlight that you can use brain models in different ways. On one end of the scale, we have data-driven models, which means that variables in the model and parameters are supposed to map onto the biological entities, sort of more or less directly to the extent possible, also quantitatively. And at the other end, we have hypothesis-driven models, or sometimes called top-down models. And there, I mean, that model can look a little bit different depending on the question you want to address. And I just wanted to point out that if we have a data-driven approach where we map model parameters directly on the biology, then it is at least easier to try to standardize the process and make it collaborative. Just to give some examples to make it more clear, if we are interested in synaptic plasticity and learning, then we want to understand how the synaptic connections changes in strength. And if we use a data-driven approach, which is also called a bottom-up approach, sometimes then we might want to simulate receptor-induced cascades to predict phosphorylation of synaptic target molecules, to predict how network activity leads to synaptic learning. In a hypothesis-driven or top-down driven approach, then we might instead pick our favorite phenomenological plasticity rule and then apply it in simulations to see if one can predict, for instance, receptor field formations or some other phenomena that we know exist in the brain. At the network level, it could be to predict neuron connectivity and network activity from single neuron features if we are using a data-driven approach, while one can rather try to mine functions of different parts of a brain structure if you use a top-down approach. So now I will exemplify what we do in Stockholm. So in Stockholm, we have several groups, both experimental groups and modeling groups, that are interested in the basal ganglia. And the basal ganglia is a forebrain structure involved in decision-making, action selection. It's also the biological substrate for reward learning. And it is of interest for many neurological and neuropsychiatric disorders, and maybe the most well-known is Parkinson's disease, of course. So the input stage of the basal ganglia is called the stratum, and it receives input from the whole cortex and thalamus. And we want to build, I will try to illustrate how we build a data-driven model of the stratum at the cellular level. So first, I should just say that the data we need is, of course, a type of neurons in the stratum, their morphology, the membrane properties, and also we have to know how many cells we have there and how they are connected. So this just is an example of how cells could look. These blue lines are the dendrites, which receive input from many other neighboring cells. And it's quite interesting that the distal parts of these dendrites, they dominate when it comes to length. And it has also been shown that they have nonlinear properties, which makes them do some information processing on their own. They can detect particular temporal patterns of inputs or particular localized inputs and so on. So then when we have the morphology, we also want to equip these model neurons with their electrical properties. And these days, this can be done in an automatized way. We use a software supporting automatized parameter estimation to try to mimic data, electrophysiological data. And this used to take like several years for a PhD student, maybe 10 years ago. So then one can try to build then a piece of brain tissue. If one focus on mice, then for a cubic millimeter of the stratum, we have around 70,000 cells. And this is how it looks if you just look at the cells that have their cell bodies within 100 micrometer. And then how to build the network. Then you can use the morphology and see when the axons of one neuron come close to the dendrites of neighboring neurons. And then one can assume that there are putative synapses there, connection points here in red circles. And when you have figured these things out, then it's time to actually use principles that you have extracted to actually predict where are the synapses. It's not in all these possible places, but there are rules for this, which now are sort of, these principles are now tested and explored in different brain areas to see how they can be generalized. This is just to remind that we can indeed simulate the brain. I mean, we are close. This is about exascale computing, and if we have that, we could actually model the human brain at the cellular level, at the level I just described for the stratum, but for the whole human. Of course, we don't have all the data that is needed for that, but I mean, there is a lot of initiatives around the world which is producing really data at the cellular level of the whole brain, for instance, transcriptomics data, which is valuable. So, will we then be able to understand our brains? I guess this is a key question, and what I just illustrated here is when you model the brain at the cellular level, and of course that would mean that we can understand things that relates to electrical activity and so on. But to really understand the brain, we have also to add on, especially the subcellular level, to understand how the brain adjusts to the inputs and to the surrounding when it learns and ages and so on. And I mean, it's an open question. I like this catchy citation. If the human brain were so simple that we could understand it, we would be so simple that we couldn't. But for sure, we already understand a lot. And I mean, it's really progressing fast. And at least I and many with me are really convinced that we have to do modeling and data-driven simulation to speed up the process. And now I will just finish with a movie, which just illustrates what it means to simulate the brain at the cellular level. This just shows first a neuron with all the dendrites and so on, and there you can see some inputs that makes the dendrites light up. And then, of course, the challenge came to connect the neurons based on morphology data and principles. And then we can use supercomputers as a tool, actually, to visualize the network, analyze the activity, and really speed up the understanding of the brain. This is just a small part. It's not so many cells, even though it looks many cells. So, thank you. I just wanted to say that those that are building, that are involved in building the striatal microcircuit in Stockholm, it's mainly at the Karolinska Institute, Sten Grillner, Alex Koslov, Johannes Hjorten, and so on, and also myself. But I'm also actually mainly working within the human brain project at another level, the subcellular level. Okay, thanks.
Thank you very much, Jeanette. And in the because of time constraints, I would like to hand over to Professor Victor Jirsa, who is a neurologist but uses modeling and simulation in order to provide new tools for personalized medicine. And when we think about how many patients we do have and when we want to develop such models, then this is, of course, there's a big need for new supercomputers helping us to address these challenges. So very pleased that you have attended this session. Thank you for coming.
Thank you, Katrin. Good afternoon, everyone. It's a pleasure to be here. Dear Madam Commissioner, dear organizers, thank you for inviting me. And hello, dear colleagues and guests. Yeah, I was asked to give an impression how we use high-performance computing to implement a workflow that takes us from individualized patient images, personalized medicine, through mathematics, all the way back to the patient bed in clinical applications. And I would like to do this along the example of epilepsy. And epilepsy is a key problem. 1% of the human population is touched by epilepsy, and a third of these patients are actually pharmacoresistant, which means for them the only hope that remains are invasive interventions, such as resective surgery, as an example. And beyond the suffering, there is also a large socio-economic burden upon the community to finance and support the treatment and the interventions with these patients. And I will focus on drug-resistant epilepsy. What we do as an entry point there is invasive explorations, because epilepsy expresses itself as fast oscillatory discharges that we can measure with so-called stereotactic EEG, in which we insert electrodes, as you see here in this CT image, that are about of this length, about 10 centimeters long, and they have 10 to 15 points on it that are spaced about 2-3 millimeters apart, that allows us to measure the neuroelectric activity in the brain. Yeah? Already in the beginning, the question arises, where do we have to place them? Yeah? Because we cannot place them everywhere. So we have to have already an insight in where to place them. And then here you see a three-dimensional representation of a patient's brain, in which we reconstruct the electrodes, and we are able to measure. Each point measures electric activity that expresses itself like this. This here is a seizure, lasts about 20 to 30 seconds. Pre-ictal activity, seizure onset, and then a characteristic is it starts in a particular area and then it spreads out, recruiting other areas that may or may not be epileptic. Yeah? But they may be healthy areas, but they are nevertheless being recruited. And this is when the clinical symptoms appear. The key question for the surgeon is, what is the epileptogenic zone? What is the tissue that is epileptogenic? And because this is the target for surgery and where the tissue will be extracted. And what we are doing in the Human Brain Project, we want to establish, or we have established, a workflow that gets us all the way from the patient images directly into the surgery room. And our workflow is as follows. We start with the neuroimaging of the patient, and we use to construct a geometric framework, an avatar. We start with the personalized structural brain images of the patient, and once we have the avatar, we need to equip it with mathematical equations, which are modeling the localized activity within an individual brain region. This allows us to create a functional brain. It still remains a generic brain, even though we have some personalization coming directly from these images, but we need to refine the network pathology of the patient, and this we do by, we call this sometimes fingerprinting of the patient. We are personalizing the brain and network model using latest machine learning techniques that are running on our high-performance clusters in order to personalize the brain, and then we can use this personalized brain as an independent, as an autonomous in silico platform to explore the different intervention possibilities. What does that mean? Everything of the patient, all the patient properties express themselves within the mathematical parameters. So we can change the parameters that mimics interventions on the brain model, and we can trace out parameter spaces. We perform parameter sweeps of our computational high-performance structures in order to find the best ways to intervene. We advise the surgeon, and the activity is performed in the clinical routine. Personalization occurs here through the structural images and through the machine learning that is fingerprinting the patient. I'll show you a few more details. So this is one particular patient of which I will show you some functional data, some epileptic seizure propagation data. We are using MRI, structural magnetic resonance imaging. We can extract the surfaces, cortical and subcortical of her brain. We can reconstruct that. And we can decompose it into individual brain areas. And we can extract, using so-called diffusion tensor-weighted imaging, also the connectivity. That allows us to put everything together in what I called earlier the avatar. This is a structural form defined in three-dimensional physical space where we can add in additional patient-specific features. This patient, for instance, had a hypothalamic hematoma that is biasing the parameters in the brain model, and this we can do through different means of changing the parameters. Then we use either high-dimensional models based on spiking neurons, as we heard about earlier. At the moment, we do not have the computational capacities in order to run the entire model. So we need to use some mathematical tricks that we borrow from statistical physics and nonlinear dynamics, as you know it also from thermodynamics, for instance. Thermodynamics, we have collective variables that we call heat or temperature that do not have an immediate microscopic equivalent. On the microscopic level, we have locomotion, we have oscillation, we have rotation of the molecules. But that expresses itself on a macro level through collective variables. And this type of so-called mean field approaches from statistical physics we can apply in order to obtain reduced representations. And this works pretty well. Here I have some examples from simulations, and also on the network level where we can express the spiking statistics and the dynamics through some mean field models. So that allows us then to put structure, patient-specific, together with mathematics and develop a self-contained, autonomous, in silico platform, aka a patient-specific brain model for the patient. The mathematics is represented here. We deal with differential equations, local dynamics, and the connectivity, where the large-scale connectivity from the patient comes here. At the moment, what we are doing, we are exploring hybrid approaches that are between the full spiking model and the mean field models, where we express the regions of interest by using some of the HPP tools describing the regions of interest through spiking models. Yeah? This is the current state of the art and some examples of the time series you see here. So this is a concept, this is a framework. The concepts we validate in animal models, because all the steps that I just traced out, we can redo in the mouse or in the rat. There are mouse scanners, rodent scanners, we can reconstruct the connectivity. The advantage of the animal studies is we can also go into the brain of the mouse and extract the real existing fibers. So it's not just images, yeah? But these fibers exist through tracer studies, through our colleagues at the Allen Institute. We can reconstruct the real connectivity of the mouse, and we can then provide tests through surrogate studies to demonstrate that there is actually a personalized advantage of using the, in this case, it's not a personalized connectivity, but the mouse-specific connectivity to make predictions about what is happening in the brain. That's one aspect of personalization. And the other aspect of personalization is the machine learning, where we perform probabilistic modeling, in particular Bayesian inference, in order to estimate the target that is of interest for the surgeon, namely the epileptogenic zone. Here you have an example of, this is actually the patient I showed you earlier. Her epileptogenic zone is represented in white, left and right epileptogenic zone, and the propagation zone that is not epileptogenic, but healthy tissue where the activity, though, propagates to, is represented in red here. This is what the clinician has estimated and what was performed, well, what would have been the target in the surgery. This is what is coming out of our data fitting machine learning approaches when we confront the model directly with the functional data, and we can reconstruct to quite some good degree the epileptogenic zone and develop actually a heat map, which is our deliverable to the clinician. Yeah? So here you have an example. These are the empirical data. These are the patient data with the electrodes implanted in her brain. Here you see the time series. Please note this is a very short seizure, a so-called simple seizure, where the electrode stayed on one hemisphere. The seizure stayed on one hemisphere. You see that this ball here is a little bigger. What I'm doing here is I'm integrating the energy that is in this rectangle, yeah, and then scaling up the diameter of the electrode that you see here in order to know where this discharge is actually located. Yeah? So let me zoom in so you see some more detailed seizure features, again, happening here on the right hemisphere and discharging. Please note the following also. Based on prior clinical data, the clinicians actually thought that the epileptogenic zone is located on the left hemisphere, because look how many electrodes were implanted in the left hemisphere and only 2 on the right. Yeah? During 2 weeks, this patient was implanted, and she suffered from many simple seizures, and during these 2 weeks we measured also 2 complex partial seizures, which looks like this, again starting in the under-implanted hemisphere, discharging and then propagating to the left-hand side, recruiting the temporal lobe and the left parahippocampus. Yeah? So this is what a seizure pattern looks like, and now the same thing in the simulation. After all the procedure that I demonstrated to you, the red dots represent the network nodes that are connected by the connectivity that I'm not showing here. And the good, important aspect is because we deal with structural data, we can take this avatar and we can plug in electrodes where we want. So we are not suffering from the initial bias of the functional data and the decisions the clinicians have made in the beginning. Yeah? So, but we plug in the electrodes here in the same positions as done in the patient, and we are reproducing the simple seizure on the right hemisphere. And for the same parameter set that came from the data fitting and the machine learning, this, let me see, this should be a video. Okay, doesn't matter. This should be a video, which shows essentially the discharges start on the right hemisphere, as you saw in the propagating seizure, then recruiting some of the areas in the thalamus and propagating to the left hemisphere, recruiting the same areas as you have seen in the patient data. So that worked actually quite well, but you can use it now as an in silico platform to represent different behaviors and test out the dynamic repertoire of seizures that is present specific for this one patient. In retrospective studies, we have done this now for 15 patients. By now, we are at 30 patients, yeah? But 15 patients are represented here. First of all, the same studies as with the mouse. Yeah? We can now make statements about the patient specificity, about our predictions, and the patient-specific connectivity provides the best matches with the functional data. This is a first proof of concept for personalized medicine, taking this type of HPC model-based approaches into the clinic. This is important because when you look at the surgery success rate over the last 50 years, I have only 20 years as a graph, but for the last 50 years, depending on the type of epilepsy, but if you take the average, the surgery success rate has not improved over the last 50 years. What has happened is it has become less invasive, but the success rate has not become bigger. When we compare this now with our virtual brain, patient-specific virtual brain-based approaches, compared to the surgery success rate for the patients retrospectively here, then we find that surgery success, patient being seizure-free, is more often accomplished when the discrepancy between the virtual brain predictions and what was actually executed by the clinician is small, and when there is a discrepancy, so there is a conflict, the rate of the surgery failure is significantly higher. This is proof of concept. On retrospective patients, leads us believe it's worthwhile to use this and run it through a clinical trial. And this is exactly what is happening now. So we are running effectively, it's the world's first clinical trial using autonomous brain network modeling. Starting on the 1st of January in 2019, 11 hospitals are participating that will perform in this clinical trial 400 prospective students, which means they will scan the patients, patients, and provide us with the data. We will virtualize the patients. We will provide the clinical report estimation of the epileptogenic zone, and we feed it back to the clinicians in the patient management conferences. They will use it as an additional tool in the decision-making, and it will go directly into the surgery. In fact, half of this is a blind study where half of the patients will be used and will benefit from the virtual brain prediction, and the other half of the patients will not benefit from that because we need correct statistical validation in order to demonstrate clearly the predictive value. Something I want to underline also is this is a academic, clinical, industrial partnership. We have Dassault Systèmes, a large French software company that is partner in this, that will, during the 5 years, develop a first prototype of the entire workflow and the modeling framework in order to explore the possibilities to translate it into the applications later on. This is absolutely crucial because theory needs to be carried into the clinical applications, yes, but in order to have an impact, we need an industrial partner that assures that this impact can also be maintained, made permanent in clinical life. And with that, I thank you for your attention, and I thank all my colleagues and friends that have contributed to this, and in particular, I'm very grateful to the Human Brain Project supporting this work. Thank you very much.
Thank you. That's good.
Yeah.
Thank you very much, Victor. This was really great. And we would like now to go over to the next talk. And you have heard today many eminent scientists who are leaders in their field, who are expert. But we wanted to make sure that we have also the next generation of researchers represented here. And this was easy because we have this Kristina Karpanova, a young researcher who is really doing cool research and who is applying the nest simulation and applying also machine learning to develop new theories and also to contribute to human brain understanding. So thank you, Kristina, for coming.
Thank you, Professor Ramons. Thank you for having me here. Okay. As you know, that neuroscience and machine learning speak rather different languages. Nevertheless, throughout the history of machine learning, we have taken quite a few concepts from neuroscience. Even the beginning of machine learning was inspired by biological networks. Recent examples taken from neuroscience to machine learning included deep neural networks, reinforcement learning, and so on and so forth. But while in the past few decades, neuroscience made incredible advances both in research in spatial and temporal scales, whether in cell phases, synapse connections, brain regions, and so on and so forth. Machine learning pretty much relied on various instantiations of a single method or function optimization in the training. And once I decided, once I finished my PhD and had some time in 2017, I decided to plunge head-on in the neuroscientific field. And therefore, I looked at two different types of research. One was the experimental result in different spatial and temporal scales, which included the development by the team of Professor Amundsen and others of the highly precise human brain atlas. And the other one was numerical simulations, or the basis of anatomically derived neural networks and their digital equivalent. And while searching, I found two incredible projects. One was the US Neuron project, and the second, and the one that I'm part of presenting today, was the Human Brain Project. And on the basis of anatomistic research of the brain, they have developed anatomical networks which are substituted to their digital equivalent. But I would want to show you two pictures. On the upper right side, you can see one project that has been finalized, and this is the slices of a 65-year-old slices of the brain of a 60-year-old person. And each slice is in the micromillimeter scale, or it's bigger as a hair, a part of your hair. And those are approximately 7,000 of those slices. And on the right lower hand, you can see a machine. And this machine is used to scan each of those slices and digitally reproduce the graph network of the biological neural network in incredible detail, and create its digital model of the brain in a scale and precise scale that we have never seen before. And I have also, during my search, looked to the NEST simulator, which is part of the Human Brain Project, developed by Professor Marcus Diesmann from the Jülich Neuroscience Center, which is responsible for simulation of spiking neural network models, including large-scale ones. And there's three specific things that I want to make attention to. First of all is that the NEST simulator works as a graph representation. So each neuron represents a node, and the connections between the nodes are the axons in the brain. And of course, the connections, or those axons in the code, are represented as a sending node, receiving node, a weight, and the delay of each axon. The NEST software is also represented as a two-layered system, which allows for large heterogeneous networks of point neurons to be simulated, either with one or smaller compartments. And of course, NEST conceptually evaluates the network model on an evenly spaced time grid. But more importantly, NEST is capable to be utilized in a large-scale supercomputers and take advantage of their multiple processor cores throughout the message passing interface and POSIX threads. And a very important research coming from the group of Professor Kunkyo last month presented incredible advances in the parallelization scheme of the NES simulator and how we can simulate the digital representation of the brain at an even bigger and more precise scale. But while I was searching for this tool and looking at how to understand how it works, I was installing it on our supercomputer, the V2Ho supercomputer that is in the Bulgarian Academy of Sciences. And I was playing around, I was annoying our administrators because I was taking too much space, or I was doing something with the memory requirements, and pretty much they were calling me almost every day in their office to telling me what I'm doing wrong. But that precise exercise made me think about something else. Why there's so much problem with the large-scale simulations? Why we are taking so much memory? How we can optimize the memory so we can put more neurons and more synapses in the simulation, and we can simulate better, larger brain areas. And therefore, I focus on several important details. First of all, I took as an important detail the transmission information, or how information is transmitted from neuron to neuron. And this is through the axons, and we had to consider also that those axons are connected to different neurons, and therefore they have different lengths and, of course, delays. Second of all, we had to consider the dendritic interface between the axons and the neurons, and how those electrical signals are moving to the synaptic clefts. And the synaptic clefts, of course, is the biochemical molecules that translate those signals to the cell membrane, and how we can digitally reproduce those transformation of electrical signal mediator ion channel reaction. So we had to take into consideration this, and also we had to consider which type of biological neuron functional model to simulate. And we have stopped at the Hodgkin-Huxley model, which is the main element of our digitally developed neural network. And the Hodgkin and Huxley were awarded the Nobel Prize for their framework. So the Hodgkin-Huxley model is the core mathematical framework for model biophysically based neural modeling. It's the basis for the tau functional neuron models. It can account or simulate, reproduce different types of synapses, spatial characteristics of the anatomical neurons. It can actually reproduce key biophysical properties of the action potential. And therefore, we have considered and we have absolutely developed functional equivalent of the HH model. So on the right side, you can see that each time that we need to calculate the action potential, we need to use several nonlinear dynamical equations from the Hodgkin and Huxley paper. And those equations, in fact, you can see them here, describe the change of the cell membrane potential under the influence of mediators and ionic channels. And of course, they record the electrical characteristics of the axons, their delay times, and the spikes from cell to cell. And in our representation, the equation for each cell type is pre-computed on a 1.0 millisecond time step from the moment of excitability of the cell until reaching its resting state. Now, of course, last year I also read a paper, can a neuroscientist take apart a microprocessor? And this is a very interesting article saying what are the difference between the brain and microprocessor, how the brain has certain characteristics like stochasticity, redundancy, robustness, and what are the difference between the microprocessors. But there are also several actually similarities. Each of them has basic computing units. Each of them has a connection between them representing the transference of information. And each of them is hierarchically structured to achieve a certain goal. And as you know, that as often happens, certain ideas are developed before their times, and they are later augmented and implemented under a different time and team with the correct architecture that is available today. And that is what we did for the first time in Europe with the team of Professor Markov and I. We implemented a brand new systolic pipeline processing for the simulation of digitally equivalent neural networks in an unprecedented way. And for that, we used the Intel Knights Landing processor, which has several specific characteristics that I want to mention. First of all, it has 72 cores. And it allows for over 200 threads to be utilized. But more importantly, this processor allows you to separate the processor in 4 quadrants. And as you can see here, you can separate that in 4 quadrants, and each of those quadrants can access only the memory that is near to it. And I forgot to mention that this processor also has 2 types of memory, the multi-channel DRAM memory, which is up to 16 gigabytes, and DDR memory as well. So here you can see the MCDRAM memory. And we have discovered that we can use the fast memory, the MCDRAM memory, separate the processor into quadrants to utilize an idea known from the first Cray computers, the systolic pipeline processing. And this is the first time that is done in the area of neuroscience. It's a very important discovery, and I will tell you why. But let me explain how the systolic pipeline works. Imagine that you are a hamster in a wheel, and you are constantly moving around in this wheel. So this pipeline in this processor is something like this wheel. So we start with the first quadrant. In an interval of 300 milliseconds, we record signals from all the axons known as an axon vector of a cell, of all cells in a specific group. And we bundled synapses in specific groups. And we utilize the whole MCDRAM memory of 16 gigabytes, which allow us in decimal arithmetic to fit around 4 million synapses in one memory. And of course, the synapses are addressed as sending node, receiving node, wait, and delayed. And once computed, those signals are transferred to the second quadrant on the right. Once the transfer is completed, the memory of the first quadrant is cleared, and the next batch of 300 milliseconds signals arrives. In the second quadrant, as you can see here, we compute the reaction of the cell according to the pre-computed and stored in a table values of the Hodgkin-Huxley model in the interval of 4 milliseconds. And the process is performed for all cells simultaneously for all 4 million synapses. And once it's completed, it's transferred to the third quadrant. The signals from the first quadrant are already computed, and they're simultaneously transferred to the second quadrant. Now, in the third and the fourth quadrants, as you can see over here and here, the third quadrant is responsible for the summation of potential of the cell membrane of a neuron that is triggered by the mediators and is freed from the synapses. And while computing the sum, the memory of the second quadrant is cleared, where new synapses information arrives again for the next 300 millisecond period. And at the same time, the first quadrant of synapses arrives the next period, and so on and so forth. On the calculated potential is then moved from the third quadrant to the fourth quadrant, where the only thing to be done is to check whether a spike is present. If a spike is present, the signal is sent to the outgoing axons. And this process is repeated in a systolic pipeline like a constantly running hamster in a wheel. And why is this important? During my work with the NEST simulator on the supercomputer, I saw that the more neurons and the more axons I put in the simulation, the more memory requirements we needed. And the memory requirements just exploded. And therefore, with the abilities of the KNL processor, we were able to actually work only in the fast memory. And as you can see from the results, this is the memory requirements or the simulation time when we use only the fast memory. It's incredibly fast. We are not going outside the MCD RAM memory. The communication time is incredibly decreased. But what is this blue line that is exploding? So this is once we unfortunately go outside of the MCD RAM memory. And once we go out that memory, our communication time increases, the communication is slowed down, and it's problematic. And this is done when if we haven't calculated correctly the connections between the neurons themselves. Now, there's some work needed to be done. First of all, we need to connect this research that we have established to the NEST simulation tool. And to achieve that, we need to utilize the well-known partial global area algorithm for the shared memory of the KNLs and utilize them in the NEST simulator. We need to establish the time overlapping between the calculations and refreshing in the data in the memory, and utilize them on the many nights landing processor. And finally, we are going to use a procedure similar to the message passing interface standard, and the consequences is that bundles are formed as data packets transferred in a pipeline manner to pipeline systolic manner, which achieve memory and processor to work concurrently with no bottleneck whatsoever. And there are certain outcomes and future outlook from our work. First of all, the algorithm structure showed unequivocally that the processor for the simulation need to consist of hundreds of thousands of simple processor elements, and that they need to be bundled in a group, that the partial global area algorithm need to be utilized in the shared fast memory for the connected axons. And of course that such chips have already been developed by IBM, by the Heidelberg Group, part of the Human Brain Project, University of Manchester, and other European scientific groups. And of course, from the readily available processors on the market, one can utilize the new NVIDIA Volta, released as Titan 5, which has over 5,000 CUDA cores, 600 tensor cores. And of course, we can expect in the next years some of the semiconductor companies to develop and offer on the market chips with over 16,000 simple processing cores. And I want to thank, finally, not only you for attending, but also my PhD supervisor, Professor Dimov, who sent me on this great adventure of machine learning, Professor Stoyan Markov, who sent me on this great adventure of computational neuroscience, and of course the group of Professor Marcus Disman, who heard about this research last year presented by me, encouraged me and gave me access to the deep architecture part of the PRAISE project. So thank you. Good.
Thank you very much, Christina, for this very nice talk. And at the end, I would like you to give you some insights into how supercomputing can help us to handle the big data challenge that we have in the human brain in order to better understand its organization. And you know already from the previous talks that the brain has this multi-level way of organization. So starting from the molecular level, from the level of single genes going to the single cellular level, to small microcircuits of cells, to the more systemic level and large networks which are involved in a particular function, like in language processing, for instance, and which are also so relevant when we think about patients like Victor Girza has introduced it. And the approach of the HPP, as you know, is already that we have wet lab research, empirical research, but we combine it with modeling, with theory, relying on a very rich infrastructure. But how to access all these different types of data, from the molecules and genes to the cellular level? Well, the answer is that we have to develop a human brain atlas. And the question then is, which kind of human brain atlas do we need? Well, we need a precise atlas of course, and this is an example of an application of a very precise atlas, again, where Vasco da Gama very nicely found India. So his competitor, Columbus, went into the wrong direction and did not end up in India, but found out where America is. This was also a success, but I'm not sure whether our funders, the European Commission, is so excited when we are finding out something which is so different from what is expected from us. So we need an accurate atlas, of course, to orient ourselves in the brain. And this atlas should also not only indicate the borders between areas, like the borders between different countries, but it should also consider the different aspects of brain organization. Like here in Humboldt's famous draft, where he expressed in his atlas the different levels of plants that can be found at a certain climate zone. So we would call that today a multimodal atlas. It is an atlas that integrates many different aspects of brain organization. But where to start? Well, we are starting from the cellular level of brain organization, and this is a section, a horizontal section through a human brain, in which we have identified two different areas in the frontal pole. The frontal pole is very important for our social behavior, for executive function, and the blue and the red areas, these are two areas that we have identified in histological sections of a human brain. How do we do it? Well, we are looking under the microscope how these histological sections look like. And you see here the cerebral cortex, and left and right to this green line, there are two different areas in the brain which differ a little bit in their cellular architecture. That is the way how the cells are distributed, what the shape of the cells is. And we can see tiny differences between these two areas, left and right, which are indicated here, for instance, in red. How to quantify that? Well, we have developed a method for observer-independent mapping these areas using on a mathematical measure, the Mahalanobis distance, and we can reproducibly define borders between cytoarchitectonic areas and sections of the human brain. And using this approach, we are mapping the human brain section by section, and then 3D reconstructs these human brains. And these are 3D reconstructions of 3 individual human brains, which differ slightly in their shape and in their slice. And in order to make them comparable, we all transfer them to a common stereotaxic space, superimpose these areas, and calculate so-called probabilistic maps. These are maps which quantify the variability in these areas between different subjects. And red means there's almost no variability, everything coincides, but blue, for instance, means there's a quite high intersubject variability in the localization of this area. And such information, of course, is important when you think about neurosurgery. They want to be relatively sure, or they want to quantify it somehow, which piece of tissue they are approaching or remove away. So we have calculated these probabilistic maps of the human brain, which is a very time-consuming procedure. And for one of these areas, we need approximately 1 man year or 1 woman year. So considering that we have 260 areas until now, you can imagine that's really huge effort. It really took a lot of time. And I think it's worthwhile to do it because we need the atlas on a sound biological basis, and we need to include intersubject variability as one of the most crucial features of brain organization. Between intersubject variability also determines, for instance, why some people have a normal healthy aging while other people have diseases while they are aging. Another aspect that we are addressing is a so-called molecular architecture of the human brain, showing the distribution of receptors to which neurotransmitters can bind. And neurotransmitters are key molecules for signal transduction. And here are just examples of some of these neurotransmitters which are excitatory or inhibitory or which are modulatory. And we take these molecular signatures then also to compare it with data from patients. And this is an example from a histological section from the hippocampus from a patient with Alzheimer. And an Alzheimer patient memory is being injured very early during the disease. And indeed, in the hippocampus, which is responsible for learning and memory, we see a dramatic loss of receptors in this particular brain region for this particular cholinergic receptor. And cholinergic drugs are almost the only drugs that are presently available to treat this very devastating disease. So to connect all these different types of data and modalities of data, we need, of course, to consider all the different aspects of brain organization, and we need a completely new atlas which has not been developed so far, and which is our endeavor here in the Human Brain Project, where we would like to deliver such an atlas. And in a few days and weeks, we will have a new release of this Human Brain Atlas, which we provide for free to the neuroscience community. And in this Human Brain Atlas, it is then possible to scroll around, to identify an area in which I'm interested in. I can zoom in, zoom out, I can rotate it, and everything goes on the fly. So the atlas is somewhere sitting in Lugano, and through the internet tools, we can access this atlas, search the data that is included there. And we are by far not yet finished with this atlas, but we are trying now to populate the atlas with more and more data, like probabilistic cytoarchitectonic maps, or we are also including step-by-step other data, for instance, receptor architectonic maps showing the molecular organization of the human brain. And the atlas is done in a very intuitive way, so I can start with an area, I can also start with a region in the brain, and then in a very intuitive way, I can get all the data, for instance, quantitative information of how the receptors are distributed in a certain brain region. We can then combine different receptor types. So-called receptor fingerprints can be extracted and downloaded, and all this information is then provided to the community. And then connecting to what Victor Girza has told us just some minutes ago, we are also starting to integrate dynamic data of patients, for instance, with epilepsy. And what you see here are, again, localization of electrodes. And in the next step, we would like really to show how are the dynamics in these patient brains which suffer from epilepsy. So the idea is really not only to consider healthy brain data, normal subjects data, but also brain data from patients. And the medical informatic platform, this is the part of the Human Brain Project where we are collecting data, not only from large cohorts, but also from hospitals. And I'm very proud to know that from Bulgaria, several hospitals are participating in this endeavor, are delivering data, and Bogdan Dragansky from the Medical Informatics Platform makes sure that these data are being integrated and then also combined with the Human Brain Atlas. We would like to go, of course, even into higher spatial resolutions, perhaps at the 1 micrometer scale. And this is what Jeanette Hellgren has mentioned. We need cellular data in order to come up with good models to understand the human brain. And when we analyze data on a cellular level that is on a micrometer level, we end up with very large images. So an image of just one human brain section has a size of 200,000 by 100,000 pixels. And when we sum this up for one single human brain, we end up with 2 to 3 petabytes. This is really lots of data, and when you remember from the first half of the day what is the storage capacities of the supercomputing centers in Europe, you can imagine that there are not so much brains could be localized on these tapes, because it is a huge data problem. And of course we would like to analyze the data and not just to store them on tapes forever. So we have developed now possibilities to 3D reconstruct these data on the level of single cells using deep learning. That is, we have developed methods that allow us to link one cell, which is in one section, to the remaining part of the same section, which is in the neighboring section. And this means we have to achieve an accuracy of 1 micrometer or better, while the distortions that we introduced during histology, they are unfortunately much larger, and they can range to 3-4 millimeters. Unfortunately, the videos today do not want to work, and therefore I am inviting you to our exhibition that is in the foyer, and where you can really have a look to how this atlas looks like in reality. And I think we can even more learn from the human brain when we learn more about the natural networks, how the cells are connected, how they are organized in layers. And on the right side, the movie that does not work, the right side movie shows an artificial neuronal network that also has this composition in layers. But the major, or one of the major differences is that this artificial neuronal network has only a couple of layers, maybe 100 if we speak about deep learning, but they are relatively uniform and similar to each other. While in the human brain, the layers are very different, independent where you are in the brain. And I'm convinced that these differences that we observe in human brain organization, that will help us in order to better understand how information processing is organized in the human brain, but also to develop much more powerful artificial networks. So I see that human brain research and computing are benefiting from each other. So this is a true win-win situation where neuroscientists approaching computer scientists with big data or simulation problems, we receive high computing power, but there's also a second circle. Our knowledge can inspire the development of new hardware and new software in the future, and I would hope that we have not only a deep cluster and deeper cluster, but perhaps a deeper cluster, and so on and so forth. So in my view, these two lines of research really converge and help us to solve problems. A very important prerequisite is, of course, to better understand the connections. So the connections, this is, I mean, how processing information. And for the human brain, this is very difficult to approach because of the size of the human brain and because of many problems. However, we are now in a position that we can 3D reconstruct even single axons of single nerve cells. And single axons have a size of maybe 10 micrometers. Sometimes they are even thinner. So these are the cables which are connecting the different cells and which process the information. And this is a 3D reconstruction of a piece of the so-called visual cortex of a brain, where we can see single axonal fibers. And since they are so densely located, we had to visualize only every 100th fiber and not more. But this is a flavor what can be achieved when we use supercomputing resources, GPU processors here, and combine it with neuroscience research and with new optical methods. Okay, so sometimes the brain is so big that it is not possible to analyze the whole human brain. And many of our researcher colleagues or clinicians, they analyze only small regions of interest. So for instance, for electron microscopy, you can only analyze 1 millimeter. But you would also like to include these data into the atlas. And we have developed a method, a tool here, which can be also seen outside, to include small volumes of interest and integrate it into the atlas while keeping the position that is the right one. So this is the idea. We want to ask the community to deliver their data, to connect it to the Human Brain Atlas, and also to benefit, of course, for their own research for the Human Brain Atlas. And one example of the application could be that we provide much higher resolution maps, for instance, of the subthalamic nucleus, which is a target structure for deep brain stimulation in patients with Parkinson, which cannot be treated in a different way. So this is the way how we would like to proceed and how we would like to contribute to basic neuroscience, to medicine, and to new computing technologies. Thank you for your attention. So one question to the organizers: how many minutes can we have for a break? 10 minutes? Okay, so please be back here in 10 minutes for the next session. Thank you.